Although DNA is based on a structure of nucleotides, the exact way these nucleotides are combined is unique for each organism. Therefore, once it's actually now possible to apply DNA technology in a manner that emphasizes these differences and erase the entire genome in a pattern for comparison. And that process is called as DNA fingerprinting. It's also called as DNA profiling or DNA typing, and is one of the best known tools for forensic science since the mid-80s, once it was discovered in Great Britain. So we have talked about many processes in the chapters previously in other biology classes about this process, but we will now look at this process. Let's take a brief look at some of these processes of how it will take place. Now, for a DNA fingerprinting, what you do is you sort DNA segments by size. Now, this may sound familiar, and the examples that you may have heard before was like, for example, if you have pastas of different sizes and you wanted to separate them, you can separate them out in sieves, which have different pore sizes, and you can separate out the different sized pasta. So similarly, we use that. What we use over here is a gel. A gel is this green agar that's lying on this plate, and the agar is actually composed of agarose, which is a derivative of an algae from seaweed. And it has small pores in there, which you really can't see. But those pores can actually filter the DNA segments based on its molecular weight. So, for example, if the shorter the fragment, it will be able to move at a further distance. And if it's the heavy molecular weight fragment, it will cause less distance. So, for example, up here, here is a gel that is shown to you, and I don't want to move that. So these green segments that are shown to you over here are mainly the application points for the DNA gels, and these are called as the wells. So you apply different samples of DNA in here, and then you let it run based on molecular weight, firstly, and secondly, based on charges. Notice you're applying it to the negative end and you're letting it run to the positive. Now these segments that are going through each lane are going to run based on molecular weight and its charges. So smaller DNA segments will move further and faster than larger DNA segments, and then this will then separate those segments. And once you have these segments sorted out and they are labeled and dyed, you can pretty much see visible bands that are shown up here. Comparing the bands from different lanes will indicate some corresponding similarities between a segment. So this is a shorthand version of this gel electrophoresis. Make sure you do watch the electrophoresis animation. Now let's going back to the beginning. In a population of any particular gene, you may have a long segment of DNA, and you may have the gene may be present in segments. So the first thing that you need to do is cut the required gene, and that is done by this enzyme called as a restriction enzyme. Restriction enzymes are mainly scissors that a biotechnologist will use, and it will cut fragments of DNA. So once fragments of DNA are cut, and now here's a term called RFLPs, which is the restricted frequency of long polypeptides. So you have genes of organisms. I'm going to give you an example of humans. For example, humans have 6 billion nucleotides. Out of those 6 billion nucleotides, we only have 2 billion nucleotides that code for certain genes. The other sequences are there for polymorphism. So there's segments that are there that makes no sense, but when a restriction enzyme is cut, for example, a restriction enzyme can have one of the common ones I'm going to give you an example is GATTAC. That is called as the GATTAC series. So wherever this nucleotide is present, it will cut the DNA. Now, this can be present in X number of folks around the world, and then if you cut out the segments, the chances of two segments having the same fragments is only going to be it will only be for certain individuals that are related to each other. Otherwise, they will not be related to each other. So you could get segments that are present in certain for example, families, siblings, or from mother, son, or daughter, father, and so such relationships. And if you compare those segments, you will have similarities. So in this process, the first thing is to cut it with the restriction enzyme, and here's a DNA sample shown in this test tube. Isolated DNA has been cut by restriction enzymes, and then these segments are then cleaved and applied in these wells. Now, these DNA segments are loaded into wells in a porous gel. The gel is an agarose gel that I mentioned. The gel floats in a buffer solution, which is then placed in electrodes. And when electrical current is run between these two segments, the DNA fragments move towards the positively charged particles, and also based on its segment size. Now, after gel electrophoresis is done, which is shown to you up here on the first diagram, you have the gene pool. Gene of interest has been isolated. The gene of interest has then been applied. The next step is the southern blot. Southern blot is mainly uses a bunch of paper blots that has radioactive probes in them. And once you when you soak the paper towels with the salt solutions on the gel, it will go ahead and it will stain the segments of the DNA with radioactive labeled proteins, and then you can develop these films later to determine where your segment of interest is. So that's a process called southern blotting. Southern blotting, for you to know, is a process by which DNA is radioactively labeled. The scientific applications for this process would be to understanding again of the DNA, sequencing of organisms in the DNAs. For example, here is an E. coli isolates from patient whose infections were not juice related. So the first lanes over here. Then we have the segments over here who drank the contaminated juice, and then we have the apple juice. Now, if these segments were isolated and you could compare them, you can see that the E. coli of patients can be studied based on its shorter molecular weight segments of DNA. So DNA fingerprinting is another process that you may have heard. DNA fingerprinting of criminals, or even for parentage identification, it is used. A lot of biotechnology actually uses E. coli, and we use E. coli because E. coli can be grown very easily. It only takes 20 minutes for a colony to grow in E. coli. So within 20 minutes, you can have a new colony of E. coli. It needs to eliminate endotoxins from the products, and the cells must be lysed to get the products for other organisms. So E. coli is one of the choices that is used in a lot of biotechnology experiments. DNA can be inserted into a cell by many processes. that is transformation, in which genes of the vectors are cut. You actually add a segment of the DNA and you grow that vector back in the E. coli. And I'm going to show you an animation on these two. Electrophoresis and protoplast fusion and microinjection and gene guns are all processes that we will look at briefly right now. Okay, so I just want to go back over one term over here. I think I gave you the RFLPs are actually standing for restricted fragment length polymorphisms, and these are that's the basis of why different fragments can be compared. So we'll talk more about this in class if you have questions over it. Pretty much understand that the sequence of the mutated gene is then used to diagnose by this hybridization technique, which is called as Southern blotting. Here is the scientific applications that I told you earlier. We talked about E. coli, and let's do the DNA can be inserted into a body, into a cell by transformation, as I mentioned, using a vector. Electroporation is when you use electrodes to incorporate the gene. Here is a gene therapy, microinjections, protoplast fusion, and gene guns. These are all very new techniques that are used under nanotechnology, and these are usually meant to design and manufacture small electronic circuits that are built at molecular level of matter, and these robots actually can be used to detect contamination in foods or diseases in plants, or sometimes even biological weapons. So we're not going to go a whole lot into each one of them. I just wanted you to sort of know these are terminologies. One of the things we do want to see is RNAi, something recent. RNAi stands for RNA interference, which is a promise for future gene therapy in treating cancer and viral infections. In RNAi, a double-stranded RNAs called the small interfering RNAs are used that they target a particular gene, such as a virus gene can be introduced into this cell. And the small interfering RNA molecules binds to the mRNA molecules. For example, all of these are the small interfering molecules that are binding with the mRNA molecules here, and it causes an enzymatic destruction, and then they silence the expression of a certain gene. In mice, the RNAi has been shown to inhibit, for example, the hepatitis B virus. The small interfering RNAs can also be injected into a cell to introduce introduced in a DNA vector later on, and a small DNA insert that codes the small interfering RNAi can also be used against the gene of interest that could be cloned in a DNA vector. So again, something real new is the RNAi, and I'm going to put this RNAi animation on your website too, so you can get familiar with that too. RNAi has been used a lot for the Human Genome Project. So a lot of animations that I'm going to make sure that you have in your weekly assignments to look at.